So last time we talked about the memory system, learning and memory. And so today we're going to talk about what happens with when we put stress on top of that system. And we're going to start out with a quote from Sapolsky. Haven't heard from him in a while.
Robert's early research demonstrated that stress can work on us in an even more frightening way.
Well, back when I was starting in this business, what I was focusing on was what seemed an utterly implausible idea at the time, which was chronic stress and chronic exposure to glucocorticoids could do something as unsubtle and grotesque as kill some of your brain cells.
As a PhD candidate at Rockefeller University in the early 80s, Sapolsky collaborated with his mentor, Dr. Bruce McEwen, to follow the path of stress into the brain. They subjected lab rats to chronic stress and then examined their brain cells. The team made an astonishing find. While the cells of normal rat brains have extensive branches, stressed rats' brain cells were dramatically smaller.
And what was most interesting in many ways was the part of the brain where this was happening, hippocampus. You take intro neurobiology anytime for the last 5,000 years, and what you learn is hippocampus is learning and memory.
Stress in these rats shrunk the part of their brain responsible for memory.
Stress affects memory in two ways. Chronic stress can actually change brain circuits so that we lose the capacity to remember things as we need to. Very severe acute stress can have another effect, which is often we refer to as stress makes you stupid, which is making it impossible for you in over short periods of time to remember things you know perfectly well.
We all know that phenomenon. We all know that one from back when we stressed ourselves by not getting any sleep at all. The next morning at 9 o'clock, we couldn't remember a single thing for that final exam. You take a human and stress them big time, long time, and you're going to have a hippocampus that pays the price as well.
All right. So, end of lecture. That's what happens with stress. Now we need to know a little bit more about that. So, three major targets that the stress system targets that are involved with learning and memory. The locus coeruleus is controlling norepinephrine release in our brain, and that norepinephrine in our brain is associated with our level of cognitive arousal, right? Are we alert? Are we vigilant? Are we drowsy? Are we not functioning well cognitively? And so when we release from the sympathetic nervous system, in terms of our fight or flight response to a stressor, right, that immediate sort of start onset of stress, norepinephrine is going to heighten our ability, our cognitive ability, right? And by heightening that arousal, if that stressor, I mean, that's good if it's a physical stressor because we want to try and get away from the physical stressor. But if it's a psychological stressor, right, it could actually be detrimental, right? Because then the person might ruminate or continue thinking about, right, the psychological stressor, and it continues being the stressor, right? So that increased arousal and heightened awareness could be a negative thing if we were dealing with a psychological stressor. The amygdala becomes activated in response to stress so that we associate that negative value with what's going on, right? And we'll talk about the fact that when we have glucocorticoids acting through the MR and GR receptors in the hippocampus, and we have negative emotional value in the amygdala, that those two things combine synergistically to have a very negative effect. In fact, that's probably what causes cell loss in the hippocampus, causes that cell death in the hippocampus we're talking about. And then finally in the hippocampus, we have those two glucocorticoid receptors that we talked about when we did an overview of the stresses. So we really didn't talk about, okay, well, why do we have two? What's the difference high affinity, one of them is a low affinity, and they do different things because stress has a biphasic effect on the hippocampus in that in the short term or at the onset of a stressor, we can facilitate memory formation. Again, if it's a physical, if we're using our stress system in the adaptive way that it was intended to get away from a physical stressor, remembering all the events that led up to encountering that physical stressor as well as what we do to escape it could be could help us in the future if we ever encounter the cues leading to that stressor or we encounter that stressor. However, long term, as we saw with Sapolsky, we start to decrease our ability to learn and remember. And we'll see that this affects, as he mentioned in that clip, both learning, but also retrieval of things that you've already learned. So it can interfere with both encoding and retrieval. Oh, that's the biphasic effects. All right. So just to refresh your memory on these GR and MR receptors, the GR receptors are found in the hippocampus. So this is the hippocampus here, or rat. Also found in the amygdala. So we're going to activate the amygdala in response to stress. Also found in the PDN, which is what's going to control future release. So this is our negative feedback loop. So these receptors in this area here are actually trying to shut down the future release of ACTH and corticotrophin releasing hormone here from the PDN. As opposed to the MR receptors, which are found almost exclusively in the hippocampus. Maybe a tad in the amygdala, pretty much in the hippocampus. All right, so as I mentioned, short-term stress is a good thing. This is the adaptive stress, the stressor that only lasts 30 minutes or so, right? And it appears to enhance memory formation, although sometimes the accuracy of that memory may not be good. And so I think Dr. Steinmetz has done some research in this area, looking at when we have an emotionally salient stimulus, right, that we remember details, but maybe we don't remember all the correct details, right? So this is the, I forget what the effect's called. The weapons focus effect, right, where you remember the gun, but you can't remember the face of the guy who was holding the gun, right? Thank you. So Cahill and Magal at UC Irvine did this experiment that Sapolsky talks about in your chapter, in which they had people read two stories. One story had a lot of emotional content. So one story is about a mother and her daughter going shopping, getting some ice cream, and then they go home. And then the other story, the girl gets hit by a car and they have to go to the hospital. So it's an emotional story. Similar content. One has emotion, one doesn't, right? And they found that people remember details of the emotional story much more so than the story without emotion, right? And so maybe that's not surprising because I just told you that that's going to happen. But what was interesting, what they were able to do was block norepinephrine through those beta blocker drugs that we talked about, right? So you give a beta blocker that's going to prevent some of the actions of norepinephrine, and it eliminated this enhancement, this memory enhancement. Right? So they were able to show that the memory enhancement is actually due to that increased heightened arousal, that norepinephrine burst that happens with the sympathetic nervous system at the start of the stressor. I have a quick clip to sort of show you what McCall does.
This work with rats, like this one, that doesn't know there's a platform hidden below the surface of this water tank, proved the role of adrenaline in making strong memories. The rat swings around the edge, then eventually ventures out and by chance bumps into the platform. The next day, he'll find it just a little bit faster. But watch this rat that learned where the platform was yesterday, then received a shot of adrenaline immediately afterward.
Notice that it starts out not on the edge,
Oh,
There you go.
oh, that's impressive. Adrenaline actually made this rat's brain remember better. And McGaw says the same thing happens in people. When we experience something emotional, positive or negative, our bodies release adrenaline, searing those memories into our brains more strongly.
What can you and I do right now to make sure we remember this conversation? I
Well,
can kick you.
Yeah, or I can embarrass you. Most of my research is with laboratory rats. And suppose I said all of a sudden, oh, I'm going to demonstrate to you, and I dropped about 6 rats right at your feet.
I remember, believe me, I
You
remember.
remember. Excuse me, don't sit on your lap. Excuse me, you're not supposed to be there. I think you'd remember.
All right. So, in addition to being able to block the effect with beta blockers, you can increase memory, learning and memory by giving adrenaline, which is norepinephrine, right? Just a different name for. All right. So, in the short term, the sympathetic nervous system is stimulating with this norepinephrine, enhancing memory, right? It also not only enhances memory, but it enhances all the things that contribute to memory. It enhances our sensory experience so that we're actually getting more information into our system. And I think, as I mentioned before, in addition to sensory input, sometimes under intense stressors, people actually report that time feels like time slows down because their brain is speeding up so much that they're able to make decisions much faster than they normally do, right? The amygdala becomes active and it starts stimulating memory formation through a hardwired connection into the hippocampus, right? So why do we remember those details? It's because the hippocampus is saying, hey, this is an important thing. This is a negative event. We don't want to ever experience this again. I'm going to stimulate the hippocampus to form these neural networks. There's an increase in blood glucose levels. All right, so remember we have other things that are going on in response to the stressor, like the cardiovascular system, right? And the digestive system. We're stimulating the breakdown of glucagon or glycogen into glucose, right? And we're increasing blood pressure, which means we're getting more blood flow to the brain. That only happens for the first 30 minutes or so. When we start prolonging past that adaptive stress response, we actually start to deprive the brain of glucose. We start to not have as much glucose available to the brain. The blood supply is still going, but the glucose levels drop after about 30 minutes. All right, and in addition to that, we have, in addition to all of this, right, increased energy to the brain, amygdala stimulating memory formation. We also have direct action on those GR and MR receptors. So, Yerkes-Dodson law says that as our arousal goes up, if we're doing a simple task, we continue to get better at that task. However, if it's a complicated task, we have this inverted U shape where there's a sort of level of arousal that is maximally beneficial to doing that complicated task. But then as we get really stressed, we're not able to do all of the cognitive things that we need to do for that difficult task, unlike the simple task. And so it starts to actually interfere with our performance, right? So we get this sort of inverse U function when we have complicated tasks. It's the transition to severe stress where we start to see that decrease in function. And so initially after that sort of the adaptive period where norepinephrine is beneficial, it starts to be responsible for disrupting LTP, right? If your brain is constantly in this high-go mode, right, we're being distracted, we're having all these other sensory inputs come in, and it becomes more difficult to form memories, right? And then the glucocorticoids can actually start to create long-term depression, where we're actually decreasing synaptic connectivity, decreasing connections in the brain over the long term. So as I mentioned, when we're highly aroused, our attention starts getting diverted from the learning and memory tasks that we're trying to do towards the physical stressor, or towards the psychological stressor. We start to ruminate on that, we start to worry about that, and that's going to decrease our decision making, our overall cognitive functioning in the frontal lobes. So that's going to make it more difficult to encoding. And then, as I mentioned, we also have problems with retrieval. When we think that the amygdala is sort of interfering with our ability to retrieve, that high activity in the amygdala is interfering with our ability to successfully retrieve those memories. Cushing's syndrome, one of our research papers is on next week, is really a collection. Anytime you see syndrome, it's not like disease, right? So there's a lot of different things that can cause this. But essentially, it means it's usually some sort of tumor in the pituitary and the adrenal gland, somewhere that's causing excess release of glucocorticoids. So in absence of stress, we're just getting a lot of stress hormones being released. And that causes a lot of the physical problems that we see with chronic stress, including reduced hippocampal and cerebellum volumes, which results in memory deficits. One of the things that's different, though, about this is because these high circulating levels of glucocorticoids are not happening because of a negative event, that we don't have the amygdala is not active in the same way it would be with a real stressor. These changes people typically recover from once the tumor is removed and the glucocorticoid levels go back down. So while there's reduced volume, it's the connectivity that's reduced, and not a loss of neurons that we see, which is different with psychological stressor or physical stressor. So here we can see an experiment that was done with a rat that's being exposed. So they're in their home cage, or they're in a test chamber, or they're in a test chamber with the scent of a cat, of a predator. That's going to stress the rat out. And they're having to do a spatial task, so kind of like that Morse water maze that we saw, right, where they have to swim around and find the platform. When they're in the home cage or the chamber without the stressor, their errors are low, they're good at the spatial task. When we add in the stressor, their errors go up, their cortisol levels go up, right? And this PDP is their, if we look at their EPSPs, their firing rates, they go way down, right? So cortisol high, hippocampus not firing well, making lots of errors. Right? Other things that we've found are if we artificially give steroid anti-inflammatory glucocorticoids that for over chronic periods of time, which these are sometimes given to help control autoimmune disorders. So we're actually using them as an immunosuppressant, because remember, chronic stress is going to suppress our immune system. So sometimes they're used in chronic inflammatory disorders or autoimmune, and sometimes those patients experience memory issues, and that's different than if they use NSAIDs, which are non-steroidal inflammatory, anti-inflammatory drugs. This can happen fairly rapidly, so if you're chronically administering glucocorticoids over several days, right, it's like having chronic stress levels. It's like the Cushing's syndrome, where we start to see memory of this. Now, these individuals typically recover from those because it's not like the regular stressor, it's like Cushing's syndrome, right? And so this is a hippocampal specific deficit, and so it's deficits with declarative memory as opposed to implicit memory, which we know goes through, doesn't have to go through the hippocampus. Right? And it appears to be retrieval of those memories as opposed to memory consolidation. So the presence of chronic glucocorticoids tends to interfere with the ability to retrieve memories. And it may be that when we add in the emotional component of a real psychological stressor, physical stressor with the amygdala, then we start to interfere with encoding as well. All right. So, as I mentioned, these glucocorticoids, these are steroid hormones that are binding to the GR and the MR. And the MR, the mineralocorticoid receptors, have a 10-fold higher affinity than the GR. What does that mean if I say they have a 10-fold higher affinity? What does an affinity mean? Talked about it way back when we kind of reviewed the basic principles of neuroscience and neurotransmitters, how they bind to receptors.
How easy it is, how much it wants is attracted to the molecule.
Yeah, it's the attraction, the affinity, right? And so how tightly is that going to bind, right? So if we have a little bit of glucocorticoids present, and we have MR and GR receptors, where are most of those glucocorticoid molecules going to end up? On the MR, on the high affinity. It's 10 times more attracted to that than to the GR, right? And so when we're activating these, as like a lot of steroid hormones do, this is acting through a second messenger system, and we're acting on transcription of genes. And there's interactions also with other stress hormones like norepinephrine can interact with this system as well. So it's not just in isolation. All right. So here's our optimal arousal. Here's our Yerkes-Dodson model, right? And when we have MR activation, these are the sorts of things that are happening, right? We're increasing dynorphin, opioid activity, so that's going to help with the stress-induced analgesia that we talked about. Right? It's acting on the kainate glutamate receptors to facilitate LTP. And it's acting on a couple of other areas as well, including oxytocin and vasopressin that we already have talked about, right? And so particularly this action on kainate for learning and memory is what we're talking about. This action on glutamate is associated with that enhanced attention and acquisition. Now, so we have a little bit of stress, right? We're mostly activating MR receptors, not really activating GR because they're 10 times less attracted to GR than they are to MR. However, if we have a high level of stress, at some point we activate all the MRs, right? And now it doesn't matter that they're higher affinity because they're all being stimulated. So now we start to add in GR activation. And the GR activation has the negative effects that are going to promote long-term depression. It's going to suppress neurogenesis, and it's going to suppress connections between neurons. That's the LTT. Right? And mediate some of this excitotoxicity. And we also have the amygdala now that's on board with this as well. Right? So remember, what receptors were found primarily just in the hippocampus? MRs, GRs, when they start to get activated, that's the amygdala coming into play, right? Being activated. And so now we get interference as we have more and more. It doesn't matter that the MRs are full and doing their thing. Now we're overriding that with the GR activation.
So the GR receptors are on the amygdala?
Yes. Should have put that image on here too. Right, so remember this, MR, these are our high affinity promoting learning and memory, primarily just found in the hippocampus, but once they get filled up, and now we have a big stress response, now we're activating GR in the amygdala here throughout the rest of the cortex. All right. So it's this affinity, the high affinity, low affinity, which is causing the biphasic effect on learning and memory. The high affinity stimulates memory, but once we overwhelm the high affinity and start to activate the low affinity GR, we get all the negative effects of stress on memory. When we have chronic activation of those, of the GR, we go beyond just sort of suppressing synaptic connectivity to reducing synaptic connectivity to even then losing or having some apoptosis and losing those pyramidal, those projection neurons in the hippocampus, right? So the behavioral effects then are that we're chronic stress through activation of the low affinity GR are weakening neural networks, which makes it harder to retrieve. So remember, if you think about that when we talked about last time, right? Once you form that neural network, you don't need the whole network to get activated to retrieve the memory. You just need enough cues to stimulate the network, and then you can retrieve that. Well, as we weaken that network, you need more and more cues in order to be able to retrieve that. It's harder and harder to retrieve. All right, so as we've been mentioning, let's pull this up. Long-term effects of this are that we get this atrophy, which in the beginning is transient, meaning it can come back, right? But the longer that we have sustained high levels of glucocorticoids paired with negative amygdala activity, we start to lose neurons, and that's not transient, that's permanent. We start losing those neurons. There is some pretty good evidence of neurogenesis of neurons in the hippocampus. And Sapolsky talks about that. The olfactory system is the other place in our brain that we see the production of new neurons in adulthood. We don't exactly know. It's pretty clear that we have production of new neurons in the hippocampus in adult developed brains. We don't exactly know what it is that those things are doing, but presumably they're helping with memory, with forming memories, being that it's in the hippocampus, and that's the primary job of the hippocampus. And so that rate of neurogenesis is inhibited by the chronic sort of presence of glucocorticoids.
Can you repeat the first thing again?
Yeah. So when we have sustained levels of glucocorticoids, we're getting transient, meaning it's reversible, decreases in connections. So we get atrophy, we get the dendritic and axonal connections shrink and become less, LTD. LTP, we get more connections, strengthening connections. LTD, we decrease those connections, right? As I mentioned before, after that initial 30 minutes of higher blood glucose, chronically sustained levels of glucocorticoids is actually going to reduce the amount of glucose available in the hippocampus, in the brain in general, right? So now we're starting to injure neurons. It's sort of like the penumbra effect, right? We have them, they're susceptible, they don't have as much energy available to them, and now we're stressing them literally, right? We're stimulating from the amygdala and from the GR receptors, and that makes them vulnerable to some. Okay, so my recording of the in-class lecture stopped. I guess the battery died or something. So I'm going to pick up here with a screen capture lecture. So just finished talking about how we create a vulnerable brain, and now we have decreases in the energy, the glucose that's available, and that can result in apoptosis, not unlike sort of the penumbra effect that happens when we have a vulnerable brain following a stroke. So what are the behavioral effects of this? The behavioral effects are that it becomes harder to retrieve memories, and memories they're not erased necessarily. It just is going to require more cues in order to retrieve them, or it's going to be harder for people to retrieve them. If we look at the neuroanatomical changes, here we see in a monkey's brain, in the top part of the top picture, we see a healthy hippocampus. These are the pyramidal projection neurons. They're all organized. They're lined up and projecting in the right areas. And in a stressed monkey down here, we see fewer cells and a disorganization of those cells. So we get that atrophy of their connections. A decrease in neurogenesis and decreased blood glucose can result in a loss of cells there. All right, and so I've mentioned the amygdala plays an important role, and the amygdala is hardwired to the hippocampus. And so studies with lesioning the amygdala or severing the connections between the amygdala and the hippocampus shows that if you eliminate the hippocampus's ability, the amygdala's ability to communicate with the hippocampus, then you prevent some of these stress-induced memory deficits, and you prevent the loss of neurons in particular, right? So you have to have the high glucocorticoid levels coupled with amygdala activity in order to get that loss of neurons. And again, this is evidenced when I talked about the Cushing syndrome where we don't see loss of neurons in systems where the glucocorticoid levels are high, like Cushing syndrome, or we could talk about some of these other effects, because we don't have that negative amygdala activity. So that makes it a transient effect. So when you remove the high glucocorticoid levels, the hippocampal volume is able to recover to normal levels because we're simply recreating the synaptic connectivity that was suppressed or lost with the high circulating glucocorticoids, right? If we look at other high stress induced hippocampal reductions like PTSD or depression, those tend to be more permanent and long lasting, probably due to the neural death, where we can't have simply regrowth of dendrites and axons. So, the difference between these two is that negative emotional component associated with PTSD and depression and high glucocorticoid activity. So as I mentioned, there are synthetic glucocorticoids like hydrocortisone, methazone, prednisone, and these are classic meds that are used to treat swelling of the brain after stroke. But that could actually maybe make neuronal loss worse, particularly in a vulnerable hippocampus, because now post-stroke you have a vulnerable brain, and now you're dumping glucocorticoids, and these synthetic glucocorticoids often bind with a greater affinity than normal glucocorticoids, which means we're going to be activating those GR receptors, the negative receptors, more so. Other types of conditions where you see synthetic glucocorticoids used are in AIDS-related dementia and autoimmune disorders. And again, any brain that's vulnerable, like Alzheimer's disease or elderly brains, having these high-strength synthetic glucocorticoids could negatively impact those brains. All right, so this is the image that Sapolsky showed in his video clip, and it's also in his book. And this experiment looked at, so the hippocampus, as we age, in general, shrinks in volume, and we begin to have memory deficits. That's a normal part of senile dementia, normal dementia. In animals where you remove their adrenal glands early on, we see a significant increase or reduction in the loss of neurons, if you will, because glucocorticoids are not present throughout that rat's life. And if we give 12 weeks of cortisol injections, we can mimic the loss of neurons that we see with aging. Right? So glucocorticoids, not all bad. In moderation, they are a good thing, right? They can actually act to increase neurogenesis if it's in that adaptive 30-minute short stimulation period where they're facilitating memory. However, long-term chronic levels certainly are negative. So why don't we all just get adrenalectomies and eliminate glucocorticoids? Well, in those animals where they saw the hippocampal volume didn't become reduced, they did see loss of a particular type of cell, an inhibitory interneuron. So we need glucocorticoids to do the normal good things in our body. We just need to be careful about having chronic high levels, having too much. Evidence in humans, we already talked at length about Cushing's syndrome, where we see different tumors and people with different types of Cushing's syndrome have different amounts of elevated glucocorticoids. And there's a strong negative correlation that the higher the level of glucocorticoid, the lower the hippocampal volume. So the amount of glucocorticoid matters in terms of impacting the hippocampus. Same thing with PTSD. If someone's been abused and has had multiple PTSD events, they tend to have smaller hippocampal volumes than someone who was exposed to a single PTSD event. And remember, with people that are susceptible for PTSD tend to have basal or lower circulating levels of glucocorticoids, meaning they have a bigger brake system. And what is the receptor for the brake system? Well, the receptor for the brake system is the GR receptor in the PVN. So if you have more GR receptors in the brain, your hippocampus is going to be more sensitive to those negative GR effects on memory. Again, major depression, people that have elevated glucocorticoid levels and major depression have lower hippocampal volume than people who have major depression with no elevated glucocorticoid levels. And then finally, as I mentioned earlier, as we age, our brake system gets weaker. And that means that our basal glucocorticoid levels are going to increase the older we get. And so here we see in a young brain, we have low circulating cortisol, predominantly activating the MR receptors, right? The MR receptors here, the good facilitating, enhancing LTP. And as we age, we get chronically higher levels of cortisol. So now we're going to start to act on the GR receptors and we start to see those negative memory events. So there was a 12-year longitudinal study where they separated individuals that were at the sort of onset of elderly age, at the end of middle age, transitioning into elderly, and they separated that group of subjects into people with low glucocorticoid levels and people with high glucocorticoid levels. And then they followed them for 12 years, tracked their increases in glucocorticoid levels. And the ones that had the highest levels of glucocorticoid levels had the most severe loss of hippocampal volume as measured by MRI and showed the most severe memory deficits. There's also a positive correlation between glucocorticoid levels and negative prognosis following stroke. So they looked at patients admitted to the ER following a stroke. Those that had the lowest glucocorticoid levels had the best survival rates, the best prognosis. Those that had, and as glucocorticoid levels increased, the prognosis became worse. And that's, again, because of a vulnerable brain. These glucocorticoids, high circulating glucocorticoids, can act on that vulnerable brain to cause atrophy and cell loss. All right, so that's the end of the stress and memory lecture. And make sure that you work on your research articles, and I have posted those to the Moodle page in terms of the order, and we will start with those presentations on Monday. We will talk about stress and sleep on Wednesday, And then no class on Friday. You're to work on your research presentations.